How about separation based on charge? I can do that too, and the technique I'm going to use to separate based on charge is called ion exchange chromatography. Ion exchange chromatography. Now remember, okay, separates based on charge. Now you'll remember that proteins, the amino acid side chains, right, some of them are negatively charged and some of them are positively charged, right? We all agreed that the amino acid side chains were a mixture of positive and negative charges, as well as all the others that we went through. And the ones that are negatively charged are what? Aspartate and glutamate. And the ones that are positively charged are going to be arginine, lysine, and histidine. And different proteins will have different amounts of these. And so at pH 7, or 7.0, or 7.4, some proteins are going to be positively charged, and some proteins are going to be negatively charged, depending on the relative amounts of arginine and lysine and histidine and aspartate and glutamate that they have. And so, at a particular pH, if the proteins are differently charged, I can separate them on the basis of charge. And the trick here is to use a column of cellulose or agarose that contains charged groups. I'm going to start using the letter C with a line above it as the word with. It'll save us a step, okay? With charged groups. And the two best examples of this are what are called an anion exchange resin. Now, you guys probably know this, but let's just remind ourselves. An anion exchange resin exchanges what? An anion exchange resin exchanges what? Alice, what is it going to exchange? Yeah, anions, of course. So if it's going to exchange anions, right, remember, what charge does the column have to be? Positive. So it has to be a positively charged resin. And the resin that we typically use, an example of one of these, is called DEAE. And that's diethyl aminoethane. Okay, and it's basically a bead. Here's my bead. And off of it are many, many charged groups. I'm only drawing one of them. It looks like this. N, H, okay. So, diethyl aminoethane, DEAE. Anybody here ever seen a mono Q column, right, or a Q column? That's what this is. That's basically this, okay? Now, and this would work if my protein had what charge? Negative charge. This will work for proteins that have a negative charge. I can separate them. Now, what about cation exchange? Resins. So, if I have a cation exchange resin, Santiago, what's a cation exchange resin going to exchange? Cations, of course. And so, what charge does this have to be? Negative, right? Because cats are positive, so cations are positive. So I need a negatively charged resin. And the classic negative charge resin that people used for many, many years is called carboxymethyl. Carboxymethyl, like carboxymethyl cellulose. Okay? And that, of course, is just a bead, except now its side chain is nothing more than our old friend acetate. Okay? Carboxymethyl, acetate. Nothing but acetate. Okay? Now, how do we actually get this to work? I'll show you an example. Here's a column where the beads are negatively charged. Is that anion exchange or cation exchange? Yeah, of course. This is a cation exchanger, right? Now remember we said proteins are going to have some positive groups and some negative groups. This protein has more negative charges than plus charges, so it's negative. So all this protein, all the red stuff's going to flow through. But the protein that has, in blue, that has more positive side chains than negative side chains, that's going to stick. Okay, and so this negatively charged protein flows through, but the blue one sticks. Okay, how do I get it off? How do I weaken that charge-charge interaction? Ionic strength, right? I can now take that same protein and I can do one of two things. So here's an example. Here are those three proteins. Here's a blue one, a purple one in the middle, and a red one. And you saw from before the red one flew right through, right? Because this had the wrong charge. But the blue one and the purple one, they're kind of stuck, and I want them to come off. So I change from a low salt buffer to a high salt buffer. That high salt concentration helps to shield the charges on that column. And that now allows the proteins to come off. Now, the one that has the fewest of the positive charges is going to come off the first. So this guy comes off first. That's the purple one. And then the other one comes off later. So even two positively charged proteins can separate on this column depending on the salt concentration that I use. Is there another trick that you can think of that I could use besides changing the salt concentration? What else could I change in my buffer? Right, perfect. I could change the pH. And then I could make my protein that was positively charged after initially negatively charged, and then it would come off. Fantastic. And the other technique that we can use is called affinity chromatography. Affinity chromatography. And in affinity chromatography, let me tell you, affinity chromatography is incredibly powerful. Really, really powerful. It's so powerful that you can sometimes purify your protein in one step. Okay? But it requires two things. It requires that you know a ligand of your protein. That is, you have to know your protein wants to bind to something. And it requires that you can make a column with the ligand attached. And the idea is really simple. What if I want to purify a protein that I happen to know binds to glucose? Okay, I could make a bead, and on top of that bead I could stick a bunch of glucose. Important. G here is glucose. It is not glycine. I'll say it again. G here is glucose, not glycine. And if I have a protein that likes to bind to glucose because it's maybe got a glucose binding pocket, here's my protein. I can purify this away from everything else by just taking my crude cell goo and putting it on this bead right like that. Okay? That will work fantastically well. Now, how do I get it off? If it's stuck to my bead, it's glucose binding, it's stuck to the glucose on my bead. How am I going to get it off the bead? What could I add to compete? Glucose. All I got to do is add some glucose, and that'll compete it off. Okay. Now, two examples of this. Okay, suppose I wanted to purify transcription factors. What might I want to put on the bead? Transcription factors are proteins that bind to specific DNA sequences. What should I put on the bead? DNA sequences. Okay? Now, you can suppose I want to purify a protein, but I don't know what it binds to. Could I engineer it to bind to something? So could I use genetic engineering? Engineering a ligand. Could I use cloning and genetic engineering to stick some kind of a tag onto it? Sure, what could I use? I could use a his tag, okay? A histidine tag. I could take the DNA from my protein, and I could stick a bunch of codons, we'll go through this at some point, for histidine, so that my protein, which ordinarily would be like this, now has stuck to one end of it a whole bunch of histidine residues, H, 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 H. Okay? And it turns out that histidine has a very high affinity for certain metals, like nickel. Cobalt. So I could make a nickel column, and I could add histidine. Okay? I'm sorry, I could express this protein. I've engineered this. I could express this in some other organism, like bacteria. And then I could use a nickel column to purify it off. What could I use to get it off my nickel column? Histidine, or what's the side chain of histidine? Imidazole. I could just use imidazole. Anybody know another kind of tag? Who's ever heard of GST? GST stands for glutathione S-transferase. This is a protein, a small protein, that I could stick, not so small, about 26,000 Daltons. I could stick it on the end of my protein, and now my protein will bind to glutathione. So I could make a column with glutathione on it, and my protein would stick to glutathione. When we do these tricks, we usually use these purification steps in combination. So a typical approach might be to do something like this. I could lyse the cells. I could centrifuge them. I could throw the pellet away. I could take my supernatant. And then I could do an ammonium sulfate precipitation. I could take this ammonium sulfate precipitation, and I could maybe, throw the supernatant away. I could take the pellet. I could resuspend it. I could dialyze away the ammonium sulfate. I could dialyze it into whatever buffer I happen to want to work with, right? And then I might do something like ion exchange chromatography. Okay? And I might follow where my protein was as it came off the column by following the activity. Let's say my protein was an enzyme and had something I could measure versus the fractions that came off the ion exchange column. And I might find not much activity, not much activity, not much activity, a big peak in activity, right? And then not much, not much. Clearly, what I want, the protein I want, is going to be in here because this has the activity. So what do I do now, right? And remember, these are fractions that are coming off because I'm taking my ion exchange column and I'm taking the ionic strength and I'm increasing it like this, okay? So when I get to the right ionic strength right here, this is the ionic strength where my protein eluted off the column. Okay? So now I take my fraction that I'm interested in. I want to purify it the next step. Now I might do gel filtration. Why am I doing gel filtration? Well, although the activity of my protein came off the ion exchange column here, chances are other stuff came off too. So I want to go one more step beyond that, and I want to purify my protein now by gel filtration. Okay? And now I finally have a reasonably pure protein. Reasonably pure. Okay, and I can follow this by watching as I purify it by doing two things. I can measure the specific activity. We're going to talk about that the next time. Or I could watch this by electrophoresis. So we run gel electrophoresis in a device like this. I will bring it with me the next time, and we will go through a little bit of the science behind it. But if I start out with a mess of proteins, and the one I care about is here, this might be the salt fractionation. I got rid of some of the proteins, but they're still there. And then the ion exchange, look, it got rid of a whole bunch of stuff. There's the one I want right there. Okay? Now, by size exclusion, I got rid of everything that was bigger and really littler. Now I'm almost here. And then if I had an affinity step, I might have just the single protein I wanted. And if I put all these steps on one gel, starting protein, the ammonium sulfate cut, the ion exchange, the size exclusion. Notice that size exclusion only gave me stuff about the same size, and then the affinity step, okay? Now, we're going to talk about how I follow this as it comes off these columns by measuring the protein concentration and the activity and the specific activity in the next lecture. We could monitor protein purification by electrophoresis, and we often will. If our protein has a particular biochemical function that we can measure, there's a better way. And that's by measuring something called the specific activity. Now, in order to measure specific activity, there are two things that we absolutely have to have. Okay? So in order to measure specific activity and monitor how this gets better as we purify our protein, the two things that we absolutely need to know are the activity, and I'll talk about what that is in a moment. And the second thing we need to know is the total amount of protein that we have at each step in our purification. Now, what do I mean by activity? Well, it's some measure of a chemical reaction that involves the protein function. Activity. This has to involve the protein's function. And we can define this in a whole bunch of different ways. How many people here have ever in their life used, for example, a restriction enzyme to cut a piece of DNA? Okay, so one that you probably are familiar with is this one called EcoR1. Comes from E. coli. It was one of the first restriction enzymes ever found. That's where the name comes from. E. coli, restriction enzyme 1. And when you buy it, it comes in a tube. And this tube has 10,000 units of EcoR1, and that unit tells us what the activity is. 1 unit is defined as the amount of enzyme required to cut a microgram of phage lambda DNA in 1 hour at 37 degrees in a reaction volume of 50 microliters. So that's an example of what we mean by the activity. Now, if we know the activity, right, and we're going to measure this activity in units. And the units will be different depending on what the protein does, but I've given you an example of one type of units. Now, how do I measure the protein concentration? There are a couple different ways that I can measure concentration. One way that I might choose to do it, right, total amount of protein. So I need to know protein concentration. And you know one way that I could do this right away, right? I could just measure the absorbance. Say at 280 nanomolar. Sorry, nanometers. Because we went through this already. If I plot the absorption as a function of wavelength, you know from the last lecture that I get an absorbance here that's either 280 or 276 nanometers, depending on whether or not I'm talking about tryptophan or tyrosine. We went through this the last time. And therefore, we said that the absorption of any solution was equal to epsilon times the concentration times L. L was the path length, epsilon was the extinction coefficient for the protein, and you would agree that the extinction coefficient for the protein was going to be, it would be some function, some sum, right, of the number of tryptophan residues and tyrosine residues that were in the protein. Okay? That makes sense. Nothing fancy there. Okay? Now, there's a problem, though, if I try to do that, and that will work really well once I have my protein semi-purified. But at the beginning, when I've just lysed my cells, there's a lot of DNA and RNA there, and they're going to absorb at 260 nanometers, and that's going to make it really complicated. And so clearly, although this is a great way later on, it's not such a good technique at the beginning. So I need some other techniques. And so some other techniques include dye binding assays. Once I know now what my protein concentration is and I know what my activity is, now finally I can calculate the specific activity. Because my specific activity is simply the total amount of activity divided by the total amount of protein. Now, why is that important? Let me show you a simple example with marbles, and then we'll go through one concrete example so you get the idea of it. Suppose that I was trying to purify the red marbles out of a beaker full of other marbles. Well, there are about 105 marbles in here, of which 5 are red. As I purify it, I'm going to try to keep as many of the red marbles as I can and lose as many of the non-red particles as I can. And so now I have those 5 red marbles now distributed only among something like 14 marbles instead of 105. So its relative enrichment for the red molecule has gone up. Let's do this to purify EcoR1, that enzyme that I showed you at the very beginning of the class. If I purify EcoR1, I'm going to take E. coli, and I'm going to take a particular strain of E. coli, and I'm going to purify out of it EcoR1. Okay? And I'm going to start off here then with some kind of a crude lysate. And I'm going to measure 3 things. I'm going to measure in each case the activity over the total protein. So maybe I start and I have 10,000 units of activity, right? And I measure my total protein and there's 100 milligrams of protein. So what this means then is that my specific activity, this term, specific activity, is then simply 100 units per milligram. And if I now denote the purification and I'm going to call this the fold purification. The fold purification here is just 1. Okay? Now I do step 2. I do some purification step, step 1. This might be ammonium sulfate cut. Okay, and when I do this, I end up with 7,500 units of activity. Why? Because no purification step is perfect. I lost some of my enzyme purifying it, just like you know from chemistry. When you try to purify something that you've synthesized, you don't get 100% yield. So I only got 7,500 units back. But I did that in 25 milligrams of protein. So now my specific activity has gone up to 300 units per milligram. The original lysate was 100 units per milligram. Now it's 300 units per milligram. How much purification did I get? 3-fold, right? Well, I told you ammonium sulfate wasn't a great step, so let's add a second step. Okay? Maybe the next step we'll try is ion exchange chromatography. And when I do ion exchange chromatography, I only get back 5,000 units. Boy, I just keep losing this enzyme every time I try to purify it. I'm losing a little more and losing a little more, right? But now I find this 5,000 units turns out to come out in only 2 milligrams of protein, because all the protein, right, that wasn't what I wanted, I got rid of by ion exchange chromatography. So this means that now I've got 2,500 units per milligram of protein-specific activity. And I started at 100, so how much purification did I get? 25-fold. Okay. Now I go to step 3 in my purification. Now maybe I use that gel filtration column. Okay? And again, I lose even more of my protein. Now I'm only down to 3,000 units. But look, I got rid of a whole bunch of other protein that wasn't what I wanted. Now I only have 0.1 milligrams of protein. So now my specific activity is 30,000 units per milligram. Okay? And I started at 100 units per milligram. So what's my fold purification now? 300. Okay? Which step worked the best at increasing the fold purification? Which step gave me the biggest net increase? Not in fold purification, but compared to the previous step. Well, this enriched it 3-fold. This enriched it about 8-fold. And this enriched it about 12-fold. So I'd say that last step was the winner. That was the one that really gave me the biggest jump by going from 25 to 300-fold purification.